Good afternoon to one and all. On behalf of Department of Biotechnology, Dr. NGR Educational and Research Institute, we welcoming you all for International Alumni Series Roadmap to Hybrid Research 2 4th day session. Today, we have Ms. Sharmila Ganesh with us. She has completed her BTech in Biotechnology in Dr. NGR Educational Research Institute. She has completed her Masters from University of Chester. Currently, she is pursuing her graduate teaching assistant in University of Adelaide. Dear Sharmila, live stream is yours.
Thank you. Thank you so much, ma'am. Hi, everyone. So I'm Sharmila. First of all, I would like to thank Assistant HRD Gomati ma'am, Professor Arun Kumar sir, Professor Anthony Bronson sir, and for this wonderful opportunity. And of course, I'm very much excited to be part of this International Anime series. And today's topic is going to be about tissue engineering and regenerative medicine, where I'm going to talk a bit about what is regenerative medicine, what is tissue engineering, and what are the materials we are going to use in it, and different types of clinical applications. And also, I'm going to talk about one particular application which is being currently used in clinical methodologies. So, yes, let's get started with the presentation. And yes, I'm sharing the screen now. So, as I said, this is going to be an overview of today's topic. In this, I'm going to talk about regenerative medicine, tissue engineering, cell source platforms for the cells we are going to culture, and what type of materials we are going to use for scaffold fabrication. There are also particular criteria for the scaffold fabrications, and we are going to talk about the biomaterials and application, and finally the clinical improvements and current limitations, which is going in clinical applications. So yes, what is regenerative medicine? Regenerative medicine is a broad field that stimulate and support on self-healing, where the body uses its own system, sometimes with the help of foreign biological materials, to recreate cells, rebuild tissues and organs. Regenerative medicine is nothing but a combination of different type of therapies, that is cell-based therapy, biomaterial-based therapy, and the combination of both is tissue engineering. So first we'll talk about cell-based therapy. Cell-based therapy is nothing but transplanting cells to replace or repair diseased tissue. So it's basically nothing but an autologous type of transfer. That is, we get a tissue or cells from own body, that is from particular individual, and through biopsy, and they are grown, they are cultured in vitro. The skin cells are then reprogrammed to different type of cells, that is cardiac cells or mesenchymal cells or neural cells, and they are put it back into the same patient. So the whole concept is like an autologous transfer method. And the next one is we are going to see about biomaterial-based therapy. In this biomaterial-based therapy, we are going to grow the host cell by the help of acellular matrix, which is all done in vivo. So the materials we'll be using in it are decellularized biomaterials, organic polymers, or composite ones, or synthetic biopolymers. So here there are few criteria for the biomaterials to be used for the in vivo growth of host cells. So they should be biocompatible, they should be bioresorbability, that is, they should have good pore structures to improve the vascularization, to pass on the signals, and for waste removal. And they also need to be really very less in immune reaction, and they should integrate well along with the local environment system. There are few challenges in this biomaterial based therapy. That is, they tend to break down easily when they are mechanically less in strength after the surgery, and because they are not able to take up the biological forces, sometimes they cause rejections, that is basically by immunological process, and sometimes they cause even fibrosis. So as in cell-based theory and biomaterial-based therapy, in cell-based therapy also they got few challenges, is that the limitation of donor cells is that we can get only through autographs if we are doing for the same person, and if we are going with the allogenic or heterologous transfer, then it's again going to cause immunological reaction. So to avoid these kind of challenges, there comes a technique called tissue engineering, which is nothing but a combination of cell-based therapy and biomaterial-based therapy. Yes, so the definition for tissue engineering, which is given by the US National Institute of Health, is it is a process of creating living and functional construct to repair or replace tissue or organ function lost due to age, disease, damage, or congenital defects. Tissue engineering evolved from the field of biomaterial development and refers to the practice of combining scaffolds, cells, and biologically active molecules into functional tissues. So, as I said, it's just the combination of cell-based therapy, biomaterials, along with some cocktail of growth factors. So, what are the cell sources we are going to use for tissue engineering in regenerative medicine? So, of course, it is the most important factor in tissue engineering and regenerative medicine. They are usually obtained via biopsy and then expanded in vitro culture and laboratories. So they can be from any type of sources. That is autologous. They are really best for the this type of um, regeneration, as they don't cause any immunological reaction. So, and the next one comes the allergenic, and the final one is heterologous. As I said, it can be from any cell source, so such as like embryonic stem cell, fetus-derived stem cell, adult stem cell, or induced pluripotent stem cells. And of course, stem cells are kind of major type of cells used in this uh, tissue engineering. Yeah, as I said, various stem cells, progenitor cells, and adult tissue-derived cells are widely being investigated in term. Among them, the adult-derived cells are dominant cell type used in clinic because of their ready availability. And of course, the stem cells are the frontline in term because they have an indefinite cell division potential and multiple differentiation potential. So here I have mentioned few evidences of stem cells which are being used in term. The mesenchymal stem cells isolated from various tissues. It's not only from bone marrow, they are even derived from adipose tissue, blood, and amniotic fluid. And there have been some researches on menstrual blood-derived stem cells. It has a higher proliferation rate, and it is less painless procedure to retrieve them, and it has got not any ethical issues. And there is also a recent publication on urinary stem cells, which has shown a proliferative potential and multipotent differentiation potential in development of urinary tract. So here I have mentioned few articles, few publications, and you can refer them later. So how these cells are grown in lab. So obviously they need a platform to grow in the labs. So these platforms are nothing but scaffolds. Scaffolds are like a 3D matrix for the cells to either proliferate, migrate, and differentiate so that the functional tissues can form in vitro. So the cells, they grow and they support structures called extracellular matrix, and this matrix just support the cells, and it acts as a relay station for various signaling molecules. Scientists have understood what's the signaling responses going within the cells and how do they interact with environment, and they organize into tissues and organisms which help them to recreate tissues to mend the damaged tissues or cells. So here they have used scaffolds with artificial or natural material on which the tissue is grown to mimic a biological process in vitro to replace the damaged tissue inside the body. So basically, the scaffolds are fabricated with a different type of material. They can be either from natural or from synthetic polymers, and they range from protein to plastics. And they are also seeded along with some growth factors, as I said earlier. So in some cases, the cells, the scaffold, and cell adhesion materials, bioactive materials are all mixed together at once, and they are allowed to self-assemble. Another method there is. We have an existing collagen scaffold in which the new tissues are created by stripping off human tissues which are like thrown after the surgery. So this one tends to be a most promising method for using a scaffold for human tissue, where they can use the patient own cells to bioengineer heart, liver, lung, and kidney tissue that would not be rejected by the immune system. There are a few criteria for scaffolds to be designed in tissue engineering, and they should be biocompatible. That is, they should elicit adequate response in the host patient, and they should possess a good porosity. That is, they should be um, interconnected in geometry to ensure that is highly porous structure, so that they are easy for the supply of oxygen or signaling molecules, waste removal, and the other vascularization process to go on well. And of course, as I said, the pore size should be good enough for the cell penetration and vascularization. And they need to be biodegradable substance, because as the new tissues grow, they have to be degraded in time. And regarding the mechanical performance, they have to be really good in strength, so that to avoid the biological stresses or force. And the surface properties, regarding the surface property, they should have both chemically and topographically in order to promote cellular adhesion and proliferation. So let's see what's the biomaterials for scaffold fabrication. As I said, they can be either natural one or the synthetic ones. So the natural ones, they can be protein origin biomaterial, polysaccharide origin biomaterial, and in synthetic biomaterial, they can be polymer based or ceramic based. For example, silk, collagen, and gelatin, they are basically used for skin wound healing or cartilage repair, then for bone regeneration, and etc. And in ceramic biomaterial, for example, hydroxyapatite, they are widely used for bone regeneration, as they really show good biocompatibility, and the mechanical strength of hydroxyapatite is very really high and similar to the natural bone structure. So, tissue engineering and regenerative medicine are being used in variety of clinical application. So, they are used to regenerate artificial skin, artificial bone, artificial cardiac tissues. They are used in cartilage repair, tendon replacement, bionics, tissue scaffold, cochlear implant, organ anatomy, organ transplant, plastic surgery, prosthesis, decellularization, drug delivery, and in study of cancer and many, many others. So here is a picture of a 3D structure of a scaffold, which is being used for tracheal replacement. And there's another picture which is showing an artificial urinary bladder, that is a lab grown one with the help of scaffolds. Yeah, so as I said, I'm going to talk about one particular clinical application of tissue engineering and regenerative medicine, so that it gives a better understanding about what is the process and what are the limitations about. So here I'm going to talk about, here I've mentioned about two journal articles that is about bone tissue engineering. So let's see. Bone is a calcified connective tissue, as everyone know, and it's made up of organic material composed of collagen and diverse forms of calcium phosphate such as hydroxyapatite. The outer layer has a low porosity of 10 to 30%, and it has got a high mechanical strength. The internal layer with high porosity of 50 to 90% and reduced mechanical properties. So both the layers are highly vascularized network, which plays a crucial role for supplying oxygen, nutrients, and removing waste products. These features are really, really challenging to reproduce them in vitro, making them difficult to obtain an ideal scaffold for bone tissue regeneration. Unlike other tissues, the bone has incredible capacity for regenerating, remodeling, and repairing its own injury in response to them. However, the supply of bone graft is challenging nowadays uh, because in case of large bone defect, infection, or tumor resection, where the regenerative process is exceeding the natural potential for self-healing. In that cases, the tissue engineering comes in application. We use scaffolds or 3D models to enhance the tissue regeneration. So it's very important for the scaffold to be in similar composition to that of the natural bone, and the mechanical property of the materials also plays a major role in this. For example, if the scaffold has got a really low mechanical strength, then that may result in fracture after applying them and making it unsuitable for regeneration at the area of bone defect. So the most commonly used bioceramic scaffold in clinical application and in clinical trials is a hydroxyapatite. As I said earlier, it has got a composition which is similar to that of a natural bone, and its biocompatibility and bioactivity have proven successfully to promote new bone formation in vivo. And for these reasons, Zakaria and his colleagues used composite materials of hydroxyapatite and natural hydrogel, such as collagen, to mimic the bone structure, showing an improvement in the in vivo results. Some other combination of hydroxyapatite and cytosine, alginate, gelatin, and other polymers. Basically, these are synthetic polymers. They've also been used in studies uh, to find the best bone substitute for for the clinical application. So, as I said, Zachary and colleagues they combined hydroxyapatite and then collagen to find the results of bone formation, and it was found that the combination of hydroxyapatite and collagen showed significantly better results than other type of scaffold materials. And the picture on the left hand side is nothing but the electron microscopy picture of hydroxyapatite. Yeah, so we have seen like how they are used and what are the results. And here there is in this one, I'm going to talk about a journal article which discussed about a phase one clinical trial that used a hydroxyapatite and cytosine bilayered scaffold. So here they created a bilayered scaffold by combining the HA and CS material. So basically by the method of heating and freeze drying technique to show the potential of scaffold in tissue engineering for osteochondral defect. So there are many methods for producing a scaffold that includes gas forming, electrospinning, freeze drying, and heating method. In this heating method and alternate heating method and freeze drying method, um, the cell viabilities are being increased, and the porosity of the scaffold has been increased. So that's why they have used this particular method for their findings. So here they developed the bilayered scaffold, and this was characterized by X-ray diffraction analysis, microcomputer tomography, and scanning electron microscopy. And they have also assessed the cell viability test by the NTS test and its mechanical strength. During the in vitro phase 1 study, they have demonstrated the capacity of hydroxyapatite and cytosine layer that supported the growth and degeneration of good bone marrow stromal cells, which then differentiated into osteoblasts and chondrocytes. The cell adhesion and morphology was analyzed by scanning electron microscopy, while the cell viability and proliferation were assessed by MTS test and DNA quantification. The obtained results projects the physiochemical and biological property of the developed bilayer scaffold, as they provide adequate support for the attachment uh, for the sorry one second. Yeah, for attachment, proliferation, differentiation of goat bone marrow cells into osteoblasts and chondrocytes. So the right-hand side picture is macroscopic appearance of hydroxyapatite and cytosine bilayered scaffold, and the left one is pictorial representation of significant effective of combination of hydroxy apatite and cytosine bilayered scaffold with that of normal scaffold only with hydroxyapatite. As you can see, the one with both the combination of hydroxyapatite and cytosine bilayered scaffold shows more viability than compared with the hydroxyapatite scaffold. So that means they have promoted a good amount of differentiation and proliferation of good bone marrow cells, and as they have differentiated into osteoblasts and chondrocytes in vitro studies. And the proliferation levels of good bone marrow cells have increased from day 1 to day 17 according to their results. And the left side shows the scaffold, the image of a scaffold seeded with good bone marrow cells. This is after 28 days of culture. Yes, so we are coming to the clinical improvements and limitations of tissue engineering and regenerative medicine, in particular with bone tissue engineering. So, of course, the scaffold fabrication methods are gaining more and more interest as they allow control process of pore size, interconnection, which facilitates gas diffusion, nutrition supply, waste removal, and vascularization, which is very similar to that of a natural environment. The advantage for the patient with these approaches are that they are injectable delivery systems, so they are minimally invasive procedure, avoiding surgery, and the waiting time for receiving the graft or treatment is being reduced, and they are also able to fill irregularly shaped defects. The designs for 3D models of high quality and relevance can significantly reduce the number of animals used in research as well as the failure of drug screening methodologies. And of course, there are few limitations in all uh, researchers. And in particular to this, there is the inability to reproduce a natural extracellular matrix and a well-engineered vascularized bone that mimics the natural bone and blood vessels. And there is also a limitation regarding the identification of proper cell source for model design, and in particular with the difficulty to isolate human primary cells and culture them in vitro for long term, since they are highly sensitive to culture condition, and sometimes they lost the differentiation potential after low number of passages in culture. Yeah, so we are headed to the conclusion now. And of course, as I said, tissue engineering is transforming the way we study physiology and has a great impact on development of new therapies. 3D cultures are being developed that bridges a gap between 2D culture and animal model. Of course, we need further researchers to optimize tissue engineering to be more effective in real-time applications. And yes, I would like to thank everyone for your patience and cooperation, and the management for this wonderful opportunity. Thank you.
Tissue repair, one of the hot topic in the current scenario. Thank you, dear. It was an absolutely sleeping talk, and you nailed it by giving insights about regenerative medicine, biomaterials, and its application. Even non-biological person now would have understood what is tissue regeneration after your presentation. Thank you so much, and all the best for your future endeavors. Dear audience, the link is posted in the description box and comment section. Thank you so much.